So today I'm just going to talk about how I ended up here at the University of Rochester and the RIT, and how my passion for scientific writing actually led me to where I am today. So I'm going to first start off saying that I am a native of Rochester, New York. I was born and I grew up here. I'm the only deaf person in my family. And interestingly, I have a twin sister and she's hearing. And when we were born, my parents didn't know that we were deaf, that I was deaf. But the doctor said I was fine, like I could hear just fine. But then there's the classical story of, you probably heard a parent packing parent behind the kid and trying to see if the kid can hear or not. My parents did the exact same thing, and it was interesting that my hearing twin sister would respond, but I didn't. So that's how my parents found out I was deaf, and they kept taking me to the doctor, and finally, I think I was about 6 months, 8 months old, and they finally said, yeah, Sarah had a profound hearing loss. So at that point, I was in a very unique and fortunate situation, I think, because when my parents found out that I was deaf, they immediately signed up to take sign language classes at ISD, Rochester School for the Deaf. And because of that, I feel that really helps with my own language development, and I was able to communicate very easily with my parents and my sister. So growing up, I had full access to American Sign Language, and my parents also sent me to speech therapy as well, so that I could develop skills to speak. And they did that because they wanted to give me the option. They wanted to give me the choices of whether to use spoken English or to use American Sign Language. And I really appreciate the opportunity that really afforded me for later on in life. So both my parents are very heavily immersed in science. My mom was a research scientist. She worked at the UofR for now I think almost over 30 years. My dad, he is a physician. He died when I was younger, but I had very early on in life a heavy influence on science. And I just was very fascinated by science and research and trying to understand the how and the why of everything. I remember going outside and we would go for like nature walks with my parents, and I one day found there was an entire intact skeleton of a raccoon. And I brought the entire intact skeleton home, and I identified every bone in the body. My mom was saying also, I was always trying to set up kitchen experiments at home, trying to figure out like why things would work, why things didn't. So from very early on, I had developed a strong interest in science, in research, in trying to understand how and why. Growing up, I was mainstream for the most part. I did go to Rochester School for the Deaf up until about 2nd grade, but then my parents transferred me over to the public school, and so I went to school at the Victor Central School District, and I had a sign language interpreter all throughout my years at the Victor Central School District. And when I graduated high school, I decided to go to college at St. Lawrence University, and that's in Canton, New York, very northern New York, almost into Canada. And the reason why I decided to go to St. Lawrence University as opposed to an institution where they have like other deaf students, one is mainly because of my twin sister. So my hearing twin sister really, really wanted to go to St. Lawrence University, mainly because they had an equestrian team, and my sister rides horses. And my sister and I were pretty close, and I wanted to be with my sister. So I decided, okay, fine, I'll go to St. Lawrence University. And the interesting thing about me going to St. Lawrence was I kind of went there with a naive mind, like, yes, I need sign language interpreters. I depend on them to do well in my classes. I depend on them to communicate with my professor. But when I went to St. Lawrence, just the idea of making sure I had sign language interpreters just didn't even register in my mind. I had this mentality like, it's gonna work. I don't know how it's gonna work, but I'm gonna find a way that it's going to work. So I went to St. Lawrence University, and if any of you have been up in this area of New York, it's very, very rural. Not a lot out there, just a lot of cows and the Adirondack Mountain. So the Disability Services Office at St. Lawrence University, of course, they were like, I don't know what to do. I was the first deaf person at the university, and I pretty much had to work with them and almost train them to understand how do you work with a deaf student? How do you go about finding sign language interpreters? And they ended up having to hire interpreters from Oswego. This person commuted from Oswego to Canton every single day, and that's about a 2 to 2.5 hour commute one way. But it worked. The interpreter that I had was the same interpreter that stuck with me all 4 years at St. Lawrence, and it actually worked out really well. So this was my first experience where I developed this attitude or this mindset of, it's going to work. It had to work. It's having that positive outlook. And I think this positive outlook is what helped carry me on for my future life experiences. So while I was at St. Lawrence, I did a couple of summers working at an undergraduate research program at the University of Rochester. And this was my first experience in biomedical research. And this is what helps me become actually very, very interested in research and seriously considering research as an actual future career. And it's what made me think about, oh, maybe I would like to go into like a PhD program. So I did 2 summers of undergraduate research at the University of Rochester. And it was during the summers when I first met Dr. Steve Dewhurst, who I'm sure many of you know. He's the Vice Dean of Research at the University of Rochester, and he's also the principal investigator of the postdoctoral training grant that is supporting my research. So I just bring this up because I first met him when I was an undergrad. I was only about 19 years old. And even up to this day, Dr. Steve Dewhurst has been a terrific sponsor and mentor for me. So that's another, I guess, life lesson that I learned very early on in my scientific career is that whether you know it or not, very early on, whether you're in college or graduate school, you will meet people who will turn out to be like excellent sponsors for your scientific career, whether it's 10 years or 15 years down the road. And in my case, I was so fortunate to meet Steve Dewhurst at such a young age. And he's been, even though I've never directly worked with him, like never directly worked in his lab, he's been a very good advocate for me for the last, I don't know, it's been 12 years now. So while I was at St. Lawrence, not only when I got my first thorough research experience, but also during my senior year in college, I took an introductory to neuroscience class. And they were team taught by 2 professors that I have up here. Joe Ehrlichman and Anna Esteban. I put these 2 professors up here because these are the 2 professors that really got me into neuroscience. I signed up for this introductory to neuroscience class only because I needed 1 or 2 more classes to complete my biology major. I didn't sign up for it because I thought neuroscience was the most interesting subject. or I wanted to pursue a degree in neuroscience. I just serendipitously just signed up for it to complete my biology major. But it turned out to be like the best decision I made while at St. Lawrence, because these two are the ones that made neuroscience so interesting and just captivating for me. And one of the research, or I guess subject areas that these two professors brought up during class was this. This is a picture that turned out to be a picture that defined maybe the next 10, 12 years of my research career. And I first want to ask anybody here, if anybody recognize what this is, or have any idea what this might be? Exactly. Yeah, so this is a picture of part of a hippocampus. It's actually a mouse hippocampus, not rat, but again, it's a rodent. And this is a picture that I first saw when I was an undergraduate student, and I just fell in love with it. And it was like a defining moment where like, this is what I want to study. So this picture is really a specific area within a mouse hippocampus. So if you look here, this is a brain section of the entire mouse brain. And I have a red box that's outlining the hippocampus. So you can see the hippocampus is this huge area in the brain. This pretty pink and blue picture right here is one portion of the hippocampus known as the dentate gyrus. This is a very special area in the brain where we have only recently found that it contains adult neural stem cells. And these stem cells have the ability to self-renew, proliferate, and differentiate into all the main cell types of the brain. And this is a phenomenon that nobody believed in when it was first discovered back in the 1960s and the 1970s. Everybody thought this is not real, or they thought maybe it just only existed in lower level, like evolutionary species such as rats, mice, maybe birds. But they thought there was no way it could apply to humans, or it doesn't even exist. But this is what really captivated my attention. And so I just want to give you a little bit of a brief history on how adult neurogenesis became accepted. So this is a picture of a very famous Spanish scientist known as Cajal. So he was a very well-known neuroscientist and pathologist, but he was also an artist. And he drew many hand-drawn pictures of neurons. Not digital images, not pictures, but actually hand-drawn images of neurons with very delicate arborization in the brain. And here's the guy that really pushed the frontiers of neuroscience and helped define the different cortical cell types in the brain, including the frontal cortex and the cerebellum. This guy, one of the research interests that he had was looking at neuronal regeneration following injury. He wanted to see if does the adult brain have the capacity to generate new neurons following some kind of injury or insult, disease, or trauma. And to his disappointment, he couldn't find any evidence of new neurons in the adult brain. So this is back in the early 1900s. And he's a very famous neuroscientist and pathologist, and when he made the statement, it pretty much laid the groundwork for the dogma in neuroscience that basically said no new neuron after birth. But then, in the early 1960s, there was a team of 2 neuroscientists. It was a husband-wife team named Joseph Altman and Shirley Bayer. They were interested in not neurogenesis or the creation of new neurons, but they were interested in neural behavior. And what they were trying to do was perfect a technique. And it's a technique known as tritiathymidine. So this is something that you can inject into mice or rats. And what it does is it labels a very specific time point within the cell cycle that coincides with DNA replication. So the tritiathymidine is a marker used for DNA replication as well as cell division. And so when people observe like tritiathymidine incorporation, they usually say there's been an increase in the number of new cells. So this husband-wife team, what they were really trying to do was not look at neurogenesis, but perfect the technique. And while they were doing it, they found that when they injected tritiathymidine into rats, they found, you can see in the arrow, like positive neurons in the cortex of young adult rats. And this is very, very unexpected. They were not looking for this, and they're like, So does this mean that this adult brain can create neurons? And they weren't really sure, but they published the results saying that this was a possibility. And this study was followed up not until about maybe 10 years later. It was in the early 1970s, where several other neuroscientists started doing the same thing. They were doing different injection paradigms of the same tritiathymidine to see if they could see positive neurons in the adult brain. And sure enough, they do find that there were positive cells in the adult brain of both rats as well as birds, like songbirds. So these are some of the very early studies that are suggesting that maybe the adult brain does have the capacity to create new neurons. But, and so those positive neurons that you see in the very green picture, those cells are located, these are the pink cells, that are located in a very specific area of the dentate gyrus, and it's known as the subgranular zone. And so this is what set up more research investigating how these neurostem cells are created in this area of the hippocampus, and how they proliferate, and how they differentiate into different cell types. And you can see here in the upper left-hand corner, the neural-like stem cells, the green cartoon cells, those are found in the subgranular zone. And those cells can self-renew and develop into immature neurons and eventually mature neurons that can become functional, and they can integrate into the existing neuronal circuitry. But when following the initial studies in the 1960s, 70s, and early 80s, right at 1985, there was another neuroscientist named Pascal Rakic. He's known as like the bad guy for adult neurogenesis research. He is the one that kept saying, no, this is not true, this did not happen. And he published several studies saying, all the previous studies that have been published in the 60s, the 70s, and the early 80s are not true. These new adult-born neurons don't even exist. And he was able to publish his results in very high-impact journals like Science. I mean, Science is the most high-impact journal you can publish a paper in. And he's basically saying there are no new neurons. And especially, there are no new neurons in the adult brain of primates. And so he was the one that kind of put a big halt on the growing field of adult neurogenesis. And everybody believes them. And they all, again, just abandoned the field and they didn't think about it. And they thought maybe adult neurogenesis is only relevant for mice, rodents, and birds, but not so much for primates or humans. So right at 1985, the whole field of adult neurogenesis research pretty much was abandoned, and nobody even looked at it again. So nobody looked at it again until the very late 1990s, which is almost about 20 years ago. I remember I was just about to start high school when new neuroscientists were starting to revisit the question of, does adult neurogenesis really exist? And there were a couple of researchers, well-known researchers, they published a series of studies in the late 1990s, from 1997 to 1999. This is what really set off an explosion of adult neurogenesis research, and what really convinced people saying, like, yes, adult neurogenesis does exist in the brain, and it is relevant to humans. So in this timeline, this is only like a small snapshot of what happened during the late 1990s. And I'm only putting up the highlights here. Basically what happened in 1997, 98, 99 is that researchers were finding that adult-generated neurons in the hippocampus can respond to changes in our environment. So for example, if you put a mouse or a rat in a novel environment or in an enriched environment, so for example, they have access to toys or like special bedding that's considered to be an enriched environment, they found that these mice or rats have increased number of adult-born neurons. There was also a study showing that adult neurogenesis decreases with stress, suggesting that the hippocampus plays an important role in stress. And that's why maybe when we're all like stressed out with classes and everything that we have on our to-do list, it has some negative effect on our health and our memory. They're thinking that maybe the hippocampus plays a role, and that is adversely impacting adult neurogenesis. Also, very interestingly, in 1998 was the first time when they discovered that adult-born neurons do exist in humans. This was a study that, as of today's ethical standards, would have never happened. What they did was they had patients that had cancer. I can't remember what type of cancer, but they had terminal cancer. And they were injected with BRDU as part of their treatment plan. And don't ask me how it happened, why it happened, but it did. And what they found was, indeed, in this middle picture right here, they had BRDU positive neurons. So BRDU is a marker for newly generated cells. It's very similar to tritiate thymidine, but it's not radioactive, so considered more safe. So in 98 was the first time they found new neurons in the adult human brain. And then in 1999 when they started looking into primates, and in contrast to that study that was published in 1985 seeing no neurons in primates, about almost 15 years later, they did the study again, and they actually found monkeys or primates do have adult-generated neurons. And they're also starting to find more adult-generated neurons in different areas of the brain outside the hippocampus. So this is when they first found some neurons in the prefrontal cortex. They also started making an association between the generation of new neurons in the adult brain and learning. They started to make the association that if you have an increased number of adult-made neurons, you have enhanced learning. So that was like the first time they started to relate a cellular biology phenomenon to a behavior effect. And they also found for the first time, which is my favorite, is that exercise also has a positive effect on the production of new neurons. And so that's why I like to believe is when you exercise and you feel good, and a lot of people say that exercise has a positive effect on their mood, memory, and cognition. I like to say it's because you're having more adult-generated neurons. So that's what happened in the late 1990s. And so you can see here the progression of adult neurogenesis research throughout history. So on the top left-hand schematic in the pre-1990s is basically saying no new neurons in the adult brain, doesn't even exist. But then in the late 1990s, in the lower left-hand corner, they found out, no, actually, the adult brain has the ability to create new neurons. And they found these new neurons in the dentate gyrus of the hippocampus, and another area of the brain, which I'm not going to talk about, but there's the olfactory bulb that has involved in our sense of smell. And then now the people are expanding the research to include other areas of the brain, such as the cortex, which is involved in many higher level executive functions. They're finding perhaps there are new neurons in the amygdala, which is heavily involved in emotional processing and fear conditioning, as well as the hypothalamus and other areas of the brain. So this is a very exciting field and very rapidly evolving. So these two questions are the two questions that define my research for the next almost 10 years. I was very interested in knowing, one, what is adult neurogenesis good for? Like, why does the hippocampus even have new neurons in the adult brain? Like, what is it doing? Why is it there? And the second question is, what is good or bad for adult neurogenesis? So if we think that adult neurogenesis has a positive beneficial role for learning, memory, cognition, then is there anything that we can do to manipulate this process to almost take advantage or harness the ability of neural stem cells? So with these two questions, in the early 2000s, people started designing more and more experiments to investigate these two questions. Again, this is only a snapshot, just a highlight of some of the research that happened in the early 2000s. So for example, in the early 2000s, people were, like I said before, looking at the creation of neurons outside the hippocampus, such as the cortex. They also started studying the effect of antidepressants and opiates. And this turned out to be a very interesting story. For example, with antidepressants, people were finding that if you administer mice or rats an antidepressant, such as fluoxetine, you have an increased number of new neurons in the hippocampus. And that is like very, very interesting. And they're wondering if, do these new neurons play a role in emotional processing? Does adult neurogenesis have some kind of underlying role in the etiology of depression or anxiety? So that was the first study that really set up the whole field of antidepressant in adult neurogenesis. The story behind opiates is what I will talk a little bit more about later on, because my postdoc mentor that I worked with later on, she studied neurogenesis and opiate. And she was the one that found that if you administer a rat opiate, such as heroin, cocaine, morphine, it decreases neurogenesis. And so people started studying drug abuse and how drug abuse affects neurogenesis. And then you can see here, more researchers were looking at like post-mortem human brain, and they were finding that neurogenesis is either increased or decreased in disease states such as Alzheimer's disease, Huntington's disease, stroke, seizure. And so this really paints the whole big picture and then shows you the scope of what adult neurogenesis entails and how adult neurogenesis may be an underlying factor for so many neurological and psychiatric disorders. And it gives a possible therapeutic benefit for maybe addressing these neurological and psychiatric disorders. So as you can see here, the story with the antidepressant kept evolving over time, where people started asking questions. Well, if you give antidepressants, yes, you see an increased number of new neurons. But the question became, does antidepressants require adult hippocampal neurogenesis in order to be effective? And there are some studies showing that no, adult neurogenesis is not required for the efficacy of antidepressants. But there's a whole debate going on about the role of antidepressants. It's a very, very interesting story. So to sum the whole history up here, basically, I want to say adult hippocampal neurogenesis is both structurally and functionally important. It is structurally important because within the hippocampus, it can create neurostem-like cells. And these cells can proliferate, they can self-renew, and give rise to mature neurons that can eventually integrate into the pre-existing circuitry. And most importantly, it is functionally important because it's been shown to be highly correlative with learning, memory, and mood regulation. So this became the focus of my research and became the focus of my passion for like the next 10 years. So because of this, when I graduated St. Lawrence University, I decided to come back to the U of R, and I got my PhD degree in toxicology. And the toxicology program focuses on environmental toxicology. So we look at how exposure to environmental pollution affects whatever you're interested in the body. And there was this one investigator in the Department of Environmental Medicine that I worked with, and she became my mentor. And the reason why I wanted to work with her is because she studied adult hippocampal neurogenesis. While I was in her lab, I was studying a class of environmental pollutants known as endocrine disrupting chemicals. Okay? And the one chemical that I was focusing on is the chemical with a very long name, and I'm not going to make Nikki spell it all out. So we abbreviated to CDD, or dioxin. So I was studying dioxin and how dioxin exposure interferes with the creation of new neurons in the adult brain. And my story was more, the reason why I was interested in this was because people looked at dioxin exposure during early development, and they're finding all kinds of bad things were happening. But nobody's really looked in the adult brain. Or if they did, they looked at very high level exposure that are not environmentally relevant, because we are mainly exposed to dioxin through our diet, and it's very low level, but almost daily. In the previous study, they were looking at one high dose exposure. So my research project focused on low level dioxin exposure and how it interfered with neurogenesis. So that was my PhD work at the U of R. When I finished my PhD, I was just so interested, still very interested in neurogenesis. I wanted to study this more and really focus on what are other extrinsic or intrinsic regulators of adult hippocampal neurogenesis, and how do they influence the production of new neurons. So there is a very well-known scientist in the field. Her name is Eisch. She's very well known. She is the person that discovered that opiate decrease adult neurogenesis. I wasn't necessarily interested in opiates or drug abuse. I was more interested in neurogenesis and what else might affect neurogenesis. And she was the person that I read so many of her papers as a graduate student, and I was just so enthusiastic, and she was the person I wanted to go work for, like her and only her. So I finished my PhD, and then immediately moved to Dallas, Texas. So this was a very big turning point in my life because, as you can probably see, up until this point, I've been in Rochester or upstate New York my whole life. And I've always been in an environment where interpreters were always available. They were just right there. All you had to do was ask, and I got an interpreter. I never suffered with lack of accommodation or feeling left out or left behind. This was a very new experience for me. So when I moved to Dallas, Texas, again, I had this mentality of being really naive. And I thought, oh, this is just gonna work itself out. It's just like when I moved to St. Louis, I was just, I don't know what I'm gonna do with interpreters. I don't know anyone in Dallas, Texas, I know nothing. And I think I moved to Dallas a little too naive, thinking that it would just work itself out. And it turned out to be not to be the case. So when I moved there, don't get me wrong, UT Southwestern is an excellent medical center. They do excellent research there. But when I moved there, basically they told me, we're not gonna provide interpreting. It's not our responsibility. I'm not gonna do anything for you. You have to do it yourself. So at this point, I was about 27 years old. Up until that point, I had never had to fight for an interpreter. I never had to advocate for myself because everybody else did it for me, whether it's my parents, my PI, the registrar at the graduate education office. Everybody did, fought for me except for me. And so I was actually a little embarrassed or ashamed when I moved there because I was oh man, I'm 27 years old and I don't know how to advocate for myself. I don't even know how to find an interpreter. But I had this mentality I'm gonna figure this out. And so I worked with my mentor, Eisch, who is a wonderful woman. She is I think this woman can walk on water. And she herself didn't know what to do. She never worked with a deaf scientist. But she was so positive, she was so open-minded, and we worked together to try to come up with a plan of how to find interpreters for me. So of course she did, the typical thing where I started to look up interpreting agencies in Dallas, and I'm trying to find an interpreter. And again, I'm doing all of this work myself. And I'm in a city that I've never been in before. I have no friends, I have no family, I have no support system at all. And it was difficult. It was a very personal, difficult thing that I had to do. So I found a couple of interpreters through some interpreting agencies. It didn't work out. They were not well qualified. they was just completely overwhelmed with the scientific terminology, even though I tried to sit down with them and explain to them the different terminology and give them saying it just wasn't working out. This is when luck comes into play right now, because right about the time I moved to Dallas, I had to buy a new car. So I had an old car that was like 12 years old, and my mom made me get rid of it because she was like, it's not safe anymore. I don't want you driving that car. And so I was like, okay, fine. I'll just wait until I get to Dallas and I'll buy a car there. And it just so happened that I walked into it was a Subaru dealership, because I ended up buying a Subaru. The salesman that was selling me my car, his wife is a sign language interpreter. And he was like, he told me, he's like, oh, my wife is a sign language interpreter, and they actually live like 5 minutes away from the car dealership. So he called up his wife and he's like, hey, can you please come in and just meet this person? You gotta meet this person, just because you gotta meet this person. And so the person came to the car dealership, and so I'm talking with her and I'm saying, hey, like, I'm from Rochester, New York. I just moved to Dallas, and I'm working at UT Southwestern, and I have no interpreter. Do you have any recommendations? Like, I can't find like a good, well-qualified interpreter. And she said, yeah, actually, I know of someone. I'm gonna skip over to this slide. She said, I know of someone. So she introduces me to this woman that I love. Her name is Rachel St. John. And I think a couple of you guys know her. She's a certified, ING certified interpreter, but she's also a physician at UT Southwestern Medical Center. And so I'm like, okay, but I've actually never heard of her. So I was like, okay, fine, she's worth a try, I'll just meet her. So we met over coffee at a Starbucks. And that was like, that was it. She is phenomenal. She became my designated interpreter for the next 3 or 4 years, or my entire time at UT Southwestern. And this was the first time I really understood what does it mean to have a designated interpreter. And what does it mean to have a core group of interpreters working with you? Like, I've heard of this concept before, but I didn't really truly understand like what it meant until I worked with Dr. St. John, and these are the rest of I had 2 other interpreters that worked with me on a regular basis. And it turned out to be the greatest thing. We worked together very well because we would sit down either before or after every lab meeting or seminar, and we would talk about the different terminology. We developed our own saying for the very specific term that I had. And it turned out to be a great thing. They were really good advocates for me. They had a very open mind, and they were willing to take on this difficult research subject that I was tackling. So there was a blessing for me in Dallas, where I got to work with Rachel St. John and my 2 other core team of interpreters. So while I was at UT Southwestern, I continued to study adult hippocampal neurogenesis in the context of so many different things. These are some of the things that I worked on while I was at UT Southwestern. So I was studying everything from like molecular regulator and transcription factor. I was looking at like space radiation, and I mean like real space radiation, where mice were placed on a space shuttle and they went up into space. I mean, it's like the real deal. It was a really cool project. I was also looking at how adult neurogenesis is affected in different psychiatric disorders, such as depression, anxiety, drug addiction. And it turned out to be a very productive postdoc. I had quite a few publications and very exciting. I loved my work, and it was just great. And just some pictures I just wanted to show you of my lab over the years at UT Southwestern. This right here is our symbol for the dentate gyrus because it's like a V in the hippocampus, and we have two of them. So for every picture we took, it was always like this. Yeah. So it was a great lab to be in. Again, I had a great team of interpreters. They were the reason why I even survived being at UT Southwestern. I also had a lot of fun. I put this picture up here in the lower left-hand corner because like the picture of me and my interpreter is just because not only I developed a close working relationship with them, but they also became my friend. And we developed a strong sense of trust. And I think that was also like another key aspect of what a true DI team is. It's not only your colleagues, but it's also your friends. And so we had a lot of fun together. I won several travel awards, and I was invited to give talks at several international conferences. So I was so fortunate to be able to go to Sweden, Brazil, and Australia to present my work. So towards the end of my postdoc, I was there in this postdoc for about 3.5 years. This is where I got to the point of, okay, what am I going to do? I am 31 years old, and I realized I've always been a student or a trainee. Like, I've never had a real job. Like, I was just in school my whole life. But I like it. I enjoy being a student. I enjoy being a trainee. I love being in an academic environment. And I enjoy research. But the one thing that I didn't want to do was to be a PI. I did not want to run my own lab. It was almost like my worst career nightmare. Like, I love everything about research. I just don't want to run my own lab. So I'm getting to the point where, like, what do I do? Because I love research, I love being in academia, but I don't want to run my lab. And this is when I realized, well, one of the things that I really enjoy doing, in fact, probably my favorite thing to do in the lab, is writing. So I really like being able to pull together and synthesize all the new data and all the experiments I've done and synthesize into like a story for publication. And I found myself enjoying writing more so than doing experiments at the bench. So at this point, it was a combination of knowing that even though I had a wonderful experience working with Amelia, I needed to move on. And secondly, she was also being recruited, and I knew that she was moving her lab. And she did. She ended up moving to her lab to UPenn in Philadelphia. So it was also the timing of like, I need to do something. So I was offered a job as a medical writer at a small consulting firm in Dallas. So this was a time where I really had to abandon my hippocampal research and go on and do something else. And I put this joke up here because I thought it was very applicable to me at this time. So I had to say goodbye to the hippocampus and start something new. So I started working as a medical writer at the company. And in a nutshell, I was miserable. I hated it. First of all, the job wasn't what they told me we were going to do, so that was one of the problems. Secondly, I just missed being in academia. I miss being able, just having that intellectual freedom to come up with my own project and almost like be my own boss, but yet have a mentor working with me. I miss having that mentor-mentee relationship instead of like boss-employee relationship. And I started the job, it was like in November that year. By the new year, like it was like 6 weeks later, I was already looking for a new job because I said, I can't stay here. Like I just can't stay here. And this is when I reconnected with Steve Drehurst at the U of R. So remember, at this time, I was about 31 years old, but I first met Steve when I was 19 years old. So this is an example of maintaining your connection throughout your career. And like you never know, the people that you meet when you're in college can have a huge influence on your career direction later on in life, whether it's like more than 10 years down the road. And this turned out to be the case. I mean, this is more than 10 years after I met Steve Drehurst. And I reconnected with him, and he and PJ Sinting Haidea convinced me to come back to Rochester. I am very happy that they did. They got me out of the job, and I moved back to Rochester. And so now I am, as most of you know, I'm part of a postdoctoral fellowship training program. It's a partnership between the U of R and RIT. So I split my time, most of my time is at the lab bench at the U of R, but I'm also working at the College of Health Sciences and Technology, developing some toxicology courses and teaching some of the undergraduates there. And so as far as the lab that I'm working in right now, I'm back in the Department of Environmental Medicine. It's the same department that I was in when I was a graduate student. And so I'm back to doing my toxicology route. But I'm not doing neurotoxicology anymore. Instead, our lab, or the lab that I'm in, we are focused on like maternal and fetal health and how exposure to environmental pollution can affect pregnancy outcome. So this is one graph or an example to show you that pregnant women are exposed to a wide variety of environmental pollutants. And this was a study that was, I spelled this one, but it's the National Health Assessment Survey, and it's conducted by the Centers of Disease Control, or the CDC. And it's a representative survey of the United States population. And they found that pregnant women are exposed to a wide variety of chemicals, including metal, pesticide, plastic, and it's like all over the board. And so people are interested to see how exposure to the chemical may affect maternal health as well as the health of the offspring. Our lab, we study one particular chemical known as bisphenol A, or BPA. So it's the plastic that's found in all sorts of things like Tupperware and water bottles. And we're studying how maternal exposure to BPA may influence the genetic makeup of the mom and the fetus. Not only the genetic makeup, but another phenomenon known as epigenetics, which is epigenetics is the study of different like marks or tags that get put on the DNA, and they influence the expression of genes without changing the underlying genetic sequence. So we're studying how exposure to BPA may influence the epigenetic expression and thereby may lead to a whole host of pregnancy complications, such as miscarriages, gestational diabetes, preterm birth, preeclampsia, and obesity. And all of these pathologies can have serious implications for the health of the mom and the fetus. So that's my current research today.